Hello, and welcome to today's topic. We're going to be discussing gene action from DNA to protein. So in today's lecture, what I'd like to discuss is how we go from those series of sequences in genes, the letters that we talked about in our last lecture topic, the A's, C's, T's, and G's, how we turn those into a meaningful product. And that meaningful product is actually a protein. So we're going to talk about the steps of that, including transcription and translation. We will discuss some very important things we know about the genetic code that is found in our DNA, and then we'll finish with a very brief introduction to protein folding. So up until this point, we have sort of been referring collectively to all of the DNA in our cells. However, very little of that sequence is actually dedicated to encoding a protein. So the part of our, again, collective DNA is called the genome. The part of our human genome which actually encodes for a protein is referred to as the exome. So again, the exome is a very small portion of what our genome actually codes for. A lot of our human genome controls protein synthesis. So we are looking at when proteins are made, how quickly they're made, in which locations they happen to be made. So we have a lot more responsibility in our genetic code for actually telling us when, where, and how to make these particular proteins. So our genes encode for about 20,000 different types of proteins. So again, we're a fairly, as you remember from that first lecture we talked about, we have a pretty dense genome. There's a lot of information packed into those 20,000 different proteins. So how exactly does this work? How are we turning letters into a product? So the instructions for building these is a process called protein synthesis. It involves DNA, which we're already familiar with. So many times I would refer to DNA as a set of instructions on how to build and maintain you and other living things. We're also going to include a new type of nucleic acid today called ribonucleic acid. And just as a reminder, proteins are really the jack of all trades when it comes to what's going on in our bodies. So we use proteins to do an awful lot of things. And there's a nice table here that essentially describes a whole host of different ways that we're using proteins. And this list is by no means exhaustive. There are a lot more things we're using proteins for, but this just gives you a small example. Already mentioned this, so again, all of the genetic information that we need is included in all of our cells. Okay, remember, with the exception of our sex cells, our gametes, which only contain half the genetic information we need, all of the other cells in our body have a complete set of information. However, not all cells need all of that genetic information. So the same proteins that are active in your heart are not the same proteins that are active in your liver. And the same proteins that are active in your liver are not the same proteins that are active in your skin. So we have different cell types are expressing different subsets of genes. And so this differential gene expression during the development of the fetus is actually establishing the role of the cell within the body. So very, very early on, when we have the zygote, which is when the egg and sperm have come together, and those cells start multiplying and dividing, as we continue to have more and more cells, we begin to turn on different types of genes inside of those cells. And that essentially tells the cell what it's going to be. Oh, I'm going to be a liver cell, and I'm going to be a heart cell, and I'm going to be a nerve cell. The instructions, once again, we have a set of instructions located in our DNA. But if you remember something we were talking about DNA, our chromosomes, our genes, don't leave that nuclear envelope with the exception of during the mitotic phase. That's the only time when we have the contents of our nucleus actually leaving the nucleus. Other than that, it's always housed inside that nuclear envelope. So what we need to do is we need to make a copy of the information so we can take it out to the little workbenches. If you remember, ribosomes were those little protein workbenches where we assemble proteins. we have to take a copy of the message from the nucleus out to the ribosomes where they will be assembled. To do that, we need a new type of nucleic acid, and that is where RNA comes in. So RNA is a nucleic acid, DNA is a nucleic acid, but there are some very important differences between DNA and RNA. So we've gone through DNA, right? We know it's double-stranded. The sugar is deoxyribose. We use the bases thymine, cytosine, adenine, and guanine. RNA information is a copy. We make it from a parent strand of DNA here. It's only single-stranded. We use a different type of sugar called ribose, and we use a new base called uracil. So uracil is different than thymine. So there are no T's in RNA, there are only U's. But the U has the same base pairing rule as T did. So a U will pair with A, just like we saw the A's and the T's pair together in DNA. So there are three major types of RNA that we're going to discuss: messenger RNA, ribosomal RNA, and transfer RNA. We refer to them as mRNA, rRNA, and tRNA. And each of these have a different function, and we'll go through these as we go along with today's lecture. So our messenger RNA first. It carries information that specifies a particular protein. It's produced in the nucleus. It's transported to the ribosome. And we can split it up into, let me go back up. We can split it up into 3 nucleotide sequences called codons. Most of these messenger RNAs are anywhere from 500 to 4,500 bases long. So that's the segment of the DNA that we just copied. It's typically that many bases long. The messenger RNA then is going to carry that message for us, and it has these series of codons that very specifically tells us which amino acids we're stringing together to make a protein. Our rRNA is a little bit different. It is associated with proteins, and it makes up the ribosomes themselves. Ribosomes have 2 subunits that are going to join together during protein synthesis. And rRNAs are really just structural support. So these can be anywhere from 100 to 3,000 bases long, depending on what we need. So this would be what an RNA looks like. Okay, so again, you have a larger subunit, and then you have a smaller subunit. And these are going to come together when we have the process of what we're going to call translation here in a moment as part of making a protein. Last but not least are tRNAs. tRNAs, transfer RNAs, are very short. They only have about 75 to 80 bases. And they have 2 what we're going to call working end or business ends. One end is referred to as an anticodon, which is going to form hydrogen bonds to the mRNA codon. In simple terms, a sequence of 3 letters on a tRNA is going to bond with a sequence of 3 letters on an mRNA. We refer to those regions as codons and anticodons. The other end of the tRNA is bound to, I moved that up again too fast. The other end is bound to an amino acid. So you can think of tRNAs as taxicabs. tRNA, or I guess we don't use taxis anymore. We use Lyfts and Ubers. Fine. You can think of this a tRNA as an Uber. Okay. So it's going to pick up a very specific amino acid passenger, and it's going to drop it off to a very specific place when we're going through the process of protein synthesis. Okay, so this is what a tRNA looks like. So again, you have 2 sort of working ends that are important here. This site is the site that is actually going to bind to messenger RNA, and so that we can drop off our amino acid passenger here, if you want to think of it that way. So another way to look at it is, as we're going through the process, you're going to see a series of sequences in the messenger RNA that are referred to as codons. Each of these codons corresponds to an anticodon, which has an amino acid passenger that it's going to drop off. All right, so as I mentioned before, when we're making copies, just like we saw in DNA and we made a copy of DNA, we're going to make a copy of DNA again, but this time we're using RNA to create a new molecule of RNA. So a lot of the base pairing rules are the same. We just have one new base pairing rule with RNA. Is that in DNA, the letter A is going to pair with the RNA letter U. Again, there are no T's in RNA. That's one of the ways that we can immediately distinguish the difference between a strand of DNA and a strand of RNAs. We look for the presence of thymine and uracil, and that tells us what we're looking at. So how genetic information flows. So going back to where all of that DNA is housed inside of the nucleus, all of the instructions to build and maintain you are there. However, we need to get the information out of the nucleus. That's where RNA comes into place. We're going to make a copy of the instructions. Then we're going to take those instructions and we're going to move them on to ribosomes, and those ribosomes are going to assemble proteins. So again, here's what it looks. We have two processes here, transcription and translation. Transcription is making a copy of DNA using RNA, and translation is taking that RNA, moving it out of the nucleus to a ribosome where we're going to actually assemble some proteins. All right, here's our first steps. So again, we have to go through transcription and translation. Transcription is producing a copy of the gene. Translation is decoding the copied RNA and turning it into an actual product. So let's talk about transcription first. Transcription occurs inside of the nucleus. Again, that's where the DNA is housed, so that's where the process has to take place. So we synthesize it from one strand of the DNA template, okay, which is called our template strand. The complementary strand that we have is the coding strand, okay, and it requires an enzyme called RNA polymerase. Transcription looks a lot like DNA replication, okay? So instead of a DNA polymerase enzyme, we have an RNA polymerase enzyme. So that word you should be kind of familiar with because we saw it before. And instead of copying both sides, we're not copying both sides anymore. We're just going to copy one side because that's all we really need. We don't need both sides. So when we're going through the process of transcription, okay, so we're going to go in the order. Remember, one side has to be copied backwards and the fragments were assembled when we were talking about DNA replication. We're not going to bother with any of that this time. We've just got our template strand right here, and we're just going to follow along in a continuous line until we get the entire gene that we're looking for. Next is translation. We've got the RNA. Comes out of the nucleus through those little pores in the nuclear envelope, moves it out to ribosomes. Our messenger RNA, again, can be separated into 3 letters at a time. So you can think of the words that are in our genes are 3 letters long. Every word in our gene is 3 letters long, and those words are referred to as codons, and those codons correspond to an actual thing, and those things are amino acids. The codons on the messenger RNA are going to match up with the anticodons on the transfer RNAs. The transfer RNAs pick up amino acids as passengers, and they drop them off in the corresponding correct location at the ribosome so they can be assembled into a long chain. So that's all proteins are, by the way. Proteins are long chains of amino acids, and this is how we're building them. We start the process with a start codon, which tells us it's time to begin, and we end the process with a stop codon that tells us don't add any more amino acids. We're done. All right, so if this is our messenger RNA strand here, what we have is a sequence called a Ligator sequence. This essentially is the sequence that tells us, hey, this strand of messenger RNA is ready to go. We're ready to just go ahead and start building a protein. So in order to begin translation, we have a small ribosomal subunit which will attach to our messenger RNA, and we will have the first amino acid dropped off. This is the universal start codon. It codes for an amino acid called methionine. So AUG is the codon that always tells us we're ready to go, we're ready to start the process. Once that amino acid is in place, we're going to follow along the sequence. So we're going back here to the beginning. The larger ribosomal subunit will now attach because we know we're serious. We're actually ready to go ahead and start building a product here. And so then what happens is we will continue to move down our mRNA sequence, reading it one codon at a time. Okay, and those codons then are matching up with the anticodons on the transfer RNAs that are dropping off these amino acids. So again, remember we're using these base pair rules. There are no T's, so you have A's and U's bonding together, and C's and G's still bonding together. And the process will continue then until we eventually meet a stop codon. So here's something important to note. We're talking about sort of these codons, right? These codons are what are actually responsible for telling us which amino acids go in which particular order to build these proteins. Here's the really interesting thing about our genetic code is these codons and the products that they code for are actually universal. Every living thing is using the exact same code. We are all using DNA, and our DNA and the genes inside of our DNA are using this universal genetic code, which tells us exactly which codon stands for which amino acid. So scientists have figured this out, and with those 4 combinations of letters, we can create 64 different codons. 3 of those are stop codons that don't code for an amino acid. One of them is the universal codon, right? The start AUG. All proteins start with an AUG. And then from there, we all start with methionine, and then we move on, right? So some really interesting things, and I'm pretty sure I added a genetic codon table into this lecture. If I don't have a codon table in this lecture, I will certainly post one to Blackboard at the very least, because there should be a codon table in this lecture. But if there's not, I'll add one, and I'll show you how to read it as well. Some interesting things to note about our genetic code. Our genetic code is redundant, but it's not ambiguous. So what does that mean? Redundant means there are some codons that code for the exact same amino acid, okay? So you can have 4 different codons code for lysine, for example, okay? So there are different ways to ask for the same amino acid. However, there is no ambiguity, which means that one codon cannot code for multiple amino acids. So if AUG is methionine, AUG will always be methionine, and it will never be serine or lysine or anything else. So there's never any confusion about exactly which amino acid we are needing. So again, the code's redundant, which this is going to come into play a little bit later when we start talking about mutations. The code is redundant, but it's not ambiguous. And again, it's universal. Every single living thing on this planet is using the exact same sequence of codons that use the same amino acids that we're discussing. Aha, it is here. All right. Whew. I was worried there for a minute. I thought I forgot to include an amino acid table, and I forgot how could I do that. So here is our amino acid table. This is the genetic code. So the way you read this table is the first letter of the codon is on the left, the second letter is on the top, and the third letter is on the right. So let's say I had a sequence, a codon, and it was GAU, okay? So I'd start here at the bottom, okay? This is where all of my codons that start with G are. Then I would move over to the second letter. Here's A. This is where all of my A as a second letter codons are. And then I'd finally go to the third letter, which happens to be U. Okay, it's right here at the top. Here's GAU, and it stands for aspartic acid. Okay, so I will say that you should be familiar with using this amino acid table. So if I show you a sequence of mRNA and I ask you to translate it for me, that's what this means. Translating means taking those codons and figuring out what the sequence of amino acids is. All right, so we're going to wrap things up here kind of quickly because I know that we're kind of a little bit running long here. But again, lots of great information. It can also get sort of confusing, so please feel free to play around with some of the tools that I've given you on Blackboard as additional resources because I really do think they will help in this case. A last note is that a protein is not just a sequence of amino acids. That's not what a protein is. A protein actually has to fold and take shape for it to be active and functioning and do its job. And there are technically 4 levels of protein structure. And again, we call them primary, secondary, tertiary, and quaternary. Depends on what we need to do. Its primary structure is just a series of amino acids. That's not useful. Secondary structures can be coils, an alpha helix, or a sheet, which is called a beta-pleated sheet. Our tertiary structure is when we take those coils and sheets and we begin to fold them in very unique ways. And then finally, a quaternary structure would be if you needed multiple pieces of tertiary structure put together to make a final protein. Here's what's interesting, and we'll leave it on this note. A protein can't function if a protein isn't folded the way it's supposed to fold. And typically, misfolded proteins are caused by errors in protein synthesis. So if we have the wrong amino acid in the wrong place, the protein can't fold the way it needs to fold, and it becomes useless. There's actually some disorders that humans and even some other mammals are prone to that are caused by misfolded proteins. When those misfolded proteins aggregate, they clump together and they can actually cause problems, specifically in the brain, as it turns out. So you can think of them as kind of creating a clog that your brain can't clear, and it will then present itself in different types of disorders. So again, Alzheimer's is one that probably most people are familiar with, Huntington disease, Parkinson's, a lot of these things, different types of dementia, a lot of these diseases occur over time, right? So what happens is you can have a long period of time where these proteins are misfolding and collecting in the brain, and it's not until we have a fairly large quantity of these misfolded proteins that we begin to see sort of cognitive decline. All right, so that does it for today's particular lecture. Again, please make sure you understand these terms, you know the difference between DNA, RNA, and that you can transcribe and translate a gene.